everyone, welcome back to the channel. Today we are discussing about concept of agonist, antagonist and pair receptor. For more details, check out the link in the description below. Don't forget to like and subscribe for more updates. So, first of all, what is receptor? The largest number of drugs do not bind directly to the effectors, that is enzymes, channels, transporters, structural proteins, template biomolecules, etc., but act through specific regulatory macromolecules which control the above listed effectors. These regulatory macromolecules or the sites on them which bind and interact with the drug are called receptors. So basically, when we take any medicine, it binds to the specific receptor and then show their action. This is the basic definition of receptor. Receptor is defined as a macromolecule or binding site located on the surface or inside the effector cell that recognize the signal molecule or drug and initiate the response to it, but itself has no other function. This is the example of receptor. This is GABA receptor. This is the cell membrane. The receptor is located in the transmembrane. So what is transmembrane? Means half portion of the receptor is present inside the cell and another half portion is present outside the cell. After that, these receptors have different binding sites depending on the biological molecule or intracellular biomolecules or different type of drugs. There are four types of receptor in our body. First one is ligand-gated ion channel. Second is the G-protein coupled receptors. Third is the enzyme linked receptor, and last one is the nuclear receptor. So, what is ligand-gated ion channel receptor? In ligand-gated ion channel receptors, when any drug bind to the receptor, ion channels are open, which present inside the cell, and ions are starting movement from inside to the outside. The second one is G-protein coupled receptors. The structure of G-protein coupled receptor is 7 alpha helical receptor. G-protein is bind to the receptor. When drug is bind to the receptor, receptor shows its action based on G-protein. Third type is enzyme linked receptor. So in case of enzyme linked receptor, whenever drug bind to the extracellular unit, there is an enzyme present in the intracellular unit. This enzyme activates and so their effects. Fourth type receptor is the nuclear receptor. When drugs bind to the receptor, these receptors go to the nucleus. After entering into the nucleus, it controls the transcription process. Transcription means DNA to RNA and RNA to protein synthesis. So these are the four types of receptor. Then we are discussing about all the basic definition. First is the ligand. Ligand is derived from Latin word ligar. Ligar means to bind. Any molecule which attaches selectively to particular receptors or sites. The term only indicates affinity or ability to bind without regard to functional change. Agonist and competitive antagonist are both ligands of the same receptor. It means anything it binds to the receptor, then it called ligand. Either it is endogenous molecule or any drug. Both agonist and antagonist bind to the receptor, so these are also ligands. Next definition is agonist. What is agonist? An agent which activates a receptor to produce an effect similar to that of the physiological signal molecule. For example, GABA is endogenous molecule which synthesized in our body as neurotransmitter. These neurotransmitter bind to the GABA receptor and show the effects. We are synthesizing drug barbiturate. Barbiturate also bind to the GABA receptor and show same effect as GABA. So barbiturate is agonist. Next is the inverse agonist, an agent which activates a receptor and effect in the opposite direction to that of the agonist. For example, suppose agonist bind to the receptor, activates the receptor and suppose increase the ion concentration. But inverse agonists bind to the receptor, also activate the receptor, but it decrease the ion concentration. So this is the reverse effect of agonist. Now these are the modified definition of agonist and antagonist. After modification, two terms are coming. First is the affinity and second one is the intrinsic activity. So what is affinity? The ability to bind with the receptor designated as affinity. It is scored as 0 or 1. If drug bind to the receptor, scored as 1. If drug don't bind to the receptor, scored as 0. Second term is the intrinsic activity. So what is intrinsic activity? The capacity to induce a functional change in the receptor designated as intrinsic activity. What is modified definition of agonist? Agonists have both affinity and maximal intrinsic activity. Example, adrenaline, histamine, morphine. Adrenaline is agonist of alpha and beta receptor. Morphine is opium receptor and histamine is bind to the histamine receptor. Second is the competitive antagonist. What is competitive antagonist? Competitive antagonists have affinity but no intrinsic activity. Example, chlorpheniramine, naloxone, propranolol, atropine. It means they don't change in the receptor, only bind to the receptor. They block the binding site of receptor. That's why agonists don't bind and as well as don't show their effects. After that, partial agonist. So what partial means less amount compared to the maximum amount. Partial agonists have affinity and sub-maximal intrinsic activity. Example, pentajocin. Pentajocin is partial agonist of mu opioid receptor. When it binds to mu opioid receptor, it changes in receptor. But the thing is, if opioid receptor changes in 100%, pentajocin receptor changes in 75%. Inverse agonist. Inverse agonists have affinity but intrinsic activity with a minus sign. Means it also changes in the receptor but changes happen in reverse. That's why minus sign is indicated. For example, DMCM is inverse agonist of benzodiazepine receptor. Means DMCM show opposite effect benzodiazepine. Next, we'll have different types of antagonists. Antagonist doesn't produce a biological response on binding to a receptor, but instead blocks or reduces the effect of an agonist. It may be competitive or non-competitive, means it doesn't show any cellular or biological response. So, antagonist blocks the binding site of agonist. There are two types of antagonist. First one is the competitive antagonist, and second one is the non-competitive antagonist. Competitive antagonist drugs bind selectively to a receptor without causing activation, but in such a way to prevent binding of the agonist. The antagonism may be reversible. The effect can be overcome by increasing the concentration of the agonist, will lead to the shift in the equilibrium. And non-competitive antagonist. A non-competitive antagonist may affect the reaction by binding to the active site of the receptor or to an allosteric site. Therefore, not competing with the agonist, the magnitude of the maximal response is reduced regardless of the amount of the agonist present. And after that, allosteric modulators, a drug that binds to a receptor at a site distinct from the active site. A conformational change is induced in the receptor, altering the affinity of the receptor for the endogenous ligand. We have positive allosteric modulators and also negative allosteric modulators. Positive allosteric modulators increase the affinity of the receptor for endogenous ligand. Negative allosteric modulators decrease the affinity of the receptors for the endogenous ligand. After that, we have concept pair receptors. Receptors are said to be paired when the maximal response can be elicited by an agonist at a concentration that doesn't result in 100% occupancy of available receptors. In normal language, these are the extra receptors. It has also been demonstrated that many full agonists can produce maximal response even while occupying less than 1% of available receptors. A large receptor reserve exists in their case or a number of spare receptors are present.